Okay, welcome back. This is the second installment of Chapter 15 out of Seeley dealing with the special senses. And what you're looking at here is just a diagram showing you the layout of the retina. And what we have to do is orient ourselves with the diagram here on the left. Remember that the retina is part of the neural tunic of the eyeball, and it's that portion of the eye that's capable of taking stimulus in the form of light or color and turning that into electrical information so that it can be relayed eventually back to the occipital lobe of the brain. And so what you're seeing here um, is essentially the back of the diagram is at the top of the page, and the front of the diagram here is at the bottom of the page. So light comes into the retina in this direction. You can see here where it says direction of light. What happens is that as the light comes in, passes through the cornea, then the anterior chamber filled with aqueous, then through the lens, then through the vitreous, and eventually it's going to pass through these retinal layers. First, through the ganglion layer, then the inner plexiform, then the bipolar, then the outer plexiform, then it's going to hit the photoreceptors. And after it hits the photoreceptors, it's going to be absorbed primarily by these pigmented cells. They're very dark color, and this keeps the light from bouncing around inside the eye so that photoreceptors respond only to the portion of the image that they're meant to respond to. As the light passes these photoreceptors, the cones, if they're hit with their appropriate light wavelength, be that red, green, or blue, will generate a current that will be passed from these photoreceptor cells back to the bipolars, and from the bipolars to the ganglions, and from the ganglions they're going to head straight back into the optic nerve and eventually reach the occipital lobe of the brain. The rods are very similar, except remember that the rods respond primarily to differences in light intensity and not so much changes in light wavelength. The reason for their names, rods and cones, is due to the fact that the rods have an apical layer that is rod or columnar shaped, whereas the cones have an apical layer that's shaped like an ice cream cone. Cones respond primarily to color vision, rods primarily responsible for our black and white vision. But they're wired a little bit differently to the brain. With the rods, what you have is a much broader distribution, and you have a lot more divergence in the information, the electrical information that comes from the rods that eventually gets back to the optic nerve, whereas you could think of the cones as having almost a private line to the brain, so there's not a lot of divergence in the signal. Once it's captured by the rod, it basically goes from a bipolar to a ganglion cell and straight back, instead of actually having the signal spread out in the bipolar layer so that we innervate more than one ganglion cell. And this is important because cone vision, color vision, is responsible for a lot more of the detail in our visual field than the vision information that comes from the rods. And here you can see just an electron micrograph showing you the rods and the cones and their different shapes. And the bottom line here is that when the light hits the opsin, what will happen is that it'll change its shape due to the fact that there's a molecule of retinol that's contained in these opsins. When the light hits the retinol with just the right amount of energy, what will happen is that the retinol will change its shape. It'll flip between a cis and a trans configuration, and that ultimately would change the shape of the opsin, which causes the change in the current that eventually gets sent back to the occipital lobe of the brain that we interpret as visual information in the visual cortex. So bipolar photoreceptor cells are responsible for black and white vision. They're found scattered throughout most of the retina, but not many in the fovea, and they're more sensitive to light than the cones. The protein portion that responds to light is called rhodopsin. It changes shape when it's struck by light and eventually separates into its components opsin and retinol. The retinol can be converted to vitamin A, from which it was originally made, and in the absence of light, the opsin and the retinol recombine to form rhodopsin, and at that point they're ready now for the next burst of light that comes from an image. So it's a very quick interconversion chemically. One of the unusual things about our visual field is that if we do not move our gaze slightly as we look at something, what will happen is eventually that we'll bleach out all of the rods in a particular portion of our field of vision, and we'll actually lose visual information from that part of our field of vision. It's a built-in response of the eye to move ever so slightly our gaze, even when we think we're looking in a fixed manner at an image. Our eyes are moving very, very slightly. There are records of people who can go into deep meditative states and actually bleach out the rods and go temporarily blind. But as soon as, again, we move our vision, the light hits a different population of rods in the retina and our vision returns. Rods are unusual sensory cells. When not stimulated, they're depolarized, and light causes them to actually hyperpolarize. So this is the reverse of what we think of when we think mostly of nerve and muscle cells, for instance, that depolarization produces the response. Here, hyperpolarization produces the response. The depolarization of rods causes depolarization of bipolar cells, causes depolarization of ganglion cells. The light and dark adaptation occurs as we adjust the eyes to changes in light, and this happens because of changes in the amount of rhodopsin that's around. And so what you're looking at here is just the rods and cones themselves. You can see here the outer segment, the inner segment, the nucleus, which is in the cell body, and there's the synaptic ending. Notice that the cones have a cone-shaped apical outer segment. The rods have a rod-shaped apical outer segment. And they're arranged in disc-like stacks, okay, which you can see here populating the outer membrane of these photoreceptor cells. Okay, we're zooming in here on the apical segment, and if we look at the actual membrane, what you see embedded in the membrane is the retinol and the opsin and the extracellular domain. You can see here that it's a G protein, okay, so obviously adenylate cyclase is going to be involved here, except in this case, instead of cyclic AMP, the signal is going to be cyclic GMP, which is very similar to cyclic AMP. That's the second messenger here. The G protein triggers the cyclic GMP, which is a phosphodiesterase. What's going to happen is that the phosphodiesterase will break down the cyclic GMP, and that's going to result in a change in the conformation of the gated sodium channel. So if we look at the events that produce now a visual response, here we're looking at an example from the rods, okay? So we start out at the top of the diagram here over at 1. The inactive form of retinol is cis-retinol, which means in organic chemistry terminology that the large organic groups are on the same side of a particular double bond in this long hydrocarbon chain, okay? So that's its resting state. So it's attached inside the opsin. This makes rhodopsin. Cyclic GMP is attached to the sodium channel, and this is what keeps it open. So you could think of the cyclic GMP as sort of a door jamb and the sodium channel as the door. So the cyclic GMP keeps the sodium flowing in. Light, when it hits this membrane protein, causes a shift from cis to trans retinol. This change causes opsin to change its shape from its dark to its light configuration. The activated rhodopsin activates a G protein, which activates cyclic GMP phosphodiesterase. The phosphodiesterase is going to break down the cyclic GMP into simply GMP. This causes it to diffuse away from the sodium gate. The sodium gate will then close. The all-trans retinol at this point detaches from the opsin. The G protein can then reassociate, and the cyclic GMP phosphodiesterase at this point is inactivated. What happens next is that the cis-retinol, which is still abundant in this region, will attach to the opsin. The cis-retinol essentially is formed from the trans-retinol. This returns it to its original dark configuration, and then the whole cycle can start all over again. Obviously, there are also enzymes present inside the rod cells that can continuously synthesize cyclic GMP from GTP as a precursor. Okay, so what happens here in terms of the cyclic GMP is we have. Yeah, you come in. You can have a population of cells, and you've got several intermediates here. You've got GTP as the starting material. The formation of cyclic GMP is guanylate cyclase activity. The cyclic GMP then acts as the second messenger. That keeps the sodium gate open, and it's only when the phosphodiesterase breaks down the cyclic GMP that the sodium gate will shut, and then we will cause now a change in the membrane potential. Okay, once the sodium gate shuts, the sodium doesn't come in anymore. The membrane hyperpolarizes, and that results in a change in the information. This is what we would consider to be the light signal, okay, state of that particular membrane. And again, like anything else, we have to have sufficient amounts of light in order to depolarize enough of the membrane to generate a potential that's going to eventually reach the end of the photoreceptor cell and get to the bipolar cell, causing it to fire and causing the ganglion cell in turn to fire. So here we're looking at hyperpolarization. This is the rod cell all by its lonesome. The rod cell is unstimulated in dark conditions. Rhodopsin and the G protein transducin are inactive, and the gated sodium channels are stuck open, okay, allows the sodium to enter the cell. Glutamate is constantly released from the unstimulated rod cell, and this inhibits the bipolar cells from shooting out neurotransmitter that stimulates the ganglion cells. Okay, so there's a glutamate current, this glutamate-induced current from the bipolar cells is the default state in the dark. Okay, exposure to light is going to stimulate the rods, the rhodopsin, and the attached transducin get activated. The transducin activates the phosphodiesterase. This causes the cyclic GMP to break down, to dissociate from the sodium gates. The sodium gates will then open. The sodium will then come in. Okay. What happens next is that the membrane will hyperpolarize. Okay. So this catalyzes the conversion of cyclic GMP to GMP. The sodium channels close up. Hyperpolarization takes place. This cuts off the glutamate release from the rod cells. The bipolar cells then release neurotransmitter. This stimulates the ganglion cells to make action potentials, okay? And so that's what we interpret as light vision. So it's kind of unusual. In the dark state, the rods are constantly spitting out the glutamate, and the bipolar cells are quiet, okay? And in the light state, what happens is that glutamate secretion is cut off. This induces neurotransmitter release from the bipolar cells, and that triggers a potential action potential in the ganglion cells, and that's what gets sent back to the optic nerve that we interpret as vision. In bright light, with regard to rods, more rhodopsin is broken down into vitamin A, and this protects the eye and makes it less sensitive to light. And in dark condition, more rhodopsin is produced, so the eye becomes more sensitive to light. So it takes eyes a while to accommodate when you go from dark to light and vice versa, because in part of the chemical changes that have to occur. In addition to this, we have to consider what goes on with the pupils. They constrict in bright light and they dilate in dim light, and that takes a little bit of time as well. As we age, that pupillary response slows down. That's one of the reasons why, for instance, it's difficult for older individuals to drive at night, because the pupils get and tend to stay dilated, and they're less quick to constrict when they see something bright coming on the horizon. The result is that you flood your field of vision and your acuity drops. Cones are essentially functionally very similar, but they respond to different wavelengths of light, red, green, and blue in this case. They have bipolar receptor configuration. They're responsible for color vision and visual acuity. They're very numerous in the fovea and the macula lutea, and they're fewer scattered around the rest of the retina. As the light intensity decreases, so does our ability to see color. As you probably have noticed, in dim light, it's very difficult for you to pick out yellows, reds, greens, and so forth. The visual pigment here is iodopsin. There's 3 types that respond to blue, red, and green wavelengths of light. They overlap a bit, and as a result, interpretations of color gradations are possible, so we're capable of distinguishing several million different types of visible light, okay, which are combinations of these different wavelengths. Okay, rods and cones are going to synapse with bipolar cells, which in turn synapse with ganglion cells in every area except for the fovea. In the fovea centralis, ganglion cell axons converge at the optic disc. They then exit via the optic nerve, then impulses travel to the visual cortex. The fovea centralis is our area of highest visual acuity. This is where the greatest concentration of cones will be found. Rods are responsible primarily for spatial summation. One bipolar cell gets input from a lot of different rods, and one ganglion cell gets input from several bipolar cells, and as a result, the rods don't give us a lot of visual acuity. They're primarily responsible for what you could think of as broad characteristics of the visual field, areas of high and low intensity of light as opposed to fine detail. Cones have little or no convergence. As a result, the information they provide to the brain produces high visual acuity. A receptive field is an area from which a ganglion cell gets its input, and it's roughly circular, the receptive field being at the center. Those in the fovea centralis are smaller than in other parts of the retina, and this is why there's greater visual acuity here. There's 2 types of receptive fields. An on-center ganglion cell generates more action potentials when the light is directed onto the receptive field, and it responds to light intensity, whereas an off-center neuron has more action potentials when the light is off or when the light doesn't hit the center of the field, and this responds to contrast in light. So these 2 act together to provide now our visual acuity. Interneurons present in the inner layers are there to modify the signal before it leaves the retina, heads into the optic nerve. They enhance the borders and the contours, and they increase the intensity at the borders as well. Okay, stay tuned here for a little discussion of the neuronal pathways that run from the retina to the brain. Okay, so take a listen.
Hi everyone, this is Mr. Herbst here, and today's focus is going to be on visual nerve pathways of light and images through the eye and brain. Now, that's a little bit confusing, but think of it like this. You have stuff that you see, and it has to be handled by your brain. Now, things get pretty confusing when you think about where the light goes, but let's say, for example, that you have things in your right visual field. These are things that you see in the right side of your body. They are actually handled by the left side of each corresponding eyeball. Now, why does that happen? Well, because the eye is a convex lens. So here we have the lens of the eye right here that I'm circling in red. That is a convex lens. A convex lens is thicker in the middle and thinner on the outside. So what does that do to the image? Well, here we have a convex lens. That's L right here. You may have seen this in physics, but on this side, things are coming into your eye. And on this side, this is the inside of your eye. So let's say that you're looking at a candle. The candle is right side up. Now, when it enters your eye, the convex lens is actually going to invert the image, flip it upside down. So even though you may be looking at it right side up, it's going to be upside down when it's projected on the back of your retina. Now, that's a little bit confusing to think about. So here's a diagram of pathways of light through the eye. So in blue is everything that you see on the right side of your body. So go ahead and follow the blue back. It's processed by the left side of each corresponding eye. So it's handled by this region here of each corresponding eye. That happens because of your eye lens inverts the image. Now, in your left eye, the blue, which is on the temporal side of your body, temporal means temple, is going to travel down the temporal nerve and eventually make its way to the back of the brain. Now, on this side here, something a little bit more confusing happens. This here is on the nasal side of your body. That means it's close to your nose. So your right eye, the left side of your right eye is closer to your nose. So the image is going to pass down the nasal fiber here, but it's going to have to do something weird. It's going to have to cross over at what's called the optic chiasm. So the nerve is actually going to cross over to the other side of your brain at the optic chiasm. So the left side of your brain is going to handle everything that you see on the right side of your body. However, with your left eye, it never has to cross over. But when things are processed in your right eye, things do have to cross over. The opposite happens with things that are handled by your left field of vision, right here. Follow the red back, and you follow those nasal fibers back, and they have to cross over at the optic chiasm. Things in your right field of vision are handled by the temporal nerve. Once again, they don't have to cross over. So they are going to go ahead and just travel down the temporal nerve to the right side of your visual cortex, and they're going to be handled by the brain. So let's go ahead and review that complicated mumbo-jumbo. All images are processed by opposite sides of the eye than what you may think. That happens because the lens of the eye inverts the image that you look at. There are nasal, close to the nose, and there are temporal fibers, which are close to the temple. Nasal fibers, those are the ones that are close to your nose, have to cross over at the optic chiasm, but temporal fibers do not. Here I have another picture. Let's go ahead and investigate this a little bit further. In green, I represent things that are coming in from your right field of vision, things that are on the right side of your body. Follow the green back, and they are handled by the temporal side of your left eye and the nasal side of your right eye. So the green will travel down the temporal fibers of your left eye, and it won't have to cross over, and it will go to the left side of your brain. Now, the opposite happens in your right eye. Your right eye is going to handle things from your right field of vision, but the left side, or the nasal side of your right eye, is going to be the one processing the image. So things are going to travel down, and they're going to have to cross over at the optic chiasm, and they are going to then be processed by your left visual cortex. That's the left side of your brain. Now here's where things get a little bit even more confusing. Let's say that you had a tumor that was pressing up against the temporal side of your right side of your body. Let's say that you had a tumor pressing up right against here. So it's going to ruin all those nerve synapses or those nerve connections on that side of your body. Where are you going to lose the vision in that eye? Well, follow the lines back to red over here. What handles red? The left side of your body. The right side, the temporal side of your right eye handles your left field of vision. So you are going to actually lose vision on your left side in that eye. Confusing? Sort of. Definitely confusing. Just remember that everything is opposite of what you may think. So you have a tumor pressing up on the right side or the temporal side of your right eye, you're going to actually lose vision on the left side of your body. That is a little bit confusing. Now let's go ahead and say that you had a tumor pressing up against your optic chiasm right there in the middle. And believe it or not, that can actually happen in some individuals. So all the fibers right there at the optic chiasm are going to be ruined by that tumor. Follow the nasal fibers back. So if we follow them back, Here we are in the nasal region of our right eye. What does this process? Well, that handles the right field of vision. So things on the outside of our body. Go ahead and follow this back. That handles the left field of vision. So the temporal fibers are unaffected. Those are these regions right here. These guys are still unaffected. However, these are the ones that handle stuff on the inside. So if you have a tumor pressing up against the optic chiasm, you're still going to be able to see, but you're going to have tunnel vision. Stuff that's right in the middle of your body is all you're going to be able to see. Because again, the only fibers that are unaffected are the ones that are on the temporal side of your body. Those handle stuff that's on the opposite side. So you're still going to be able to see stuff, but it's only going to be right here in the middle. You're going to be able to only see tunnel vision. You're going to lose all your peripheral vision. This stuff all will go away if there was a tumor pressing up against the optic chiasm. Anyway, this was Mr. Herbst, and this concludes the visual pathways of light through the eye and brain. I'm signing off, folks. Have a nice day.
Okay. One of the things that you may have noticed about your eyes is that they're both in the front of your head, and you might think nothing of this, but one of the advantages of having both eyes in the front of your head is that you have depth perception. The information that comes into the left and the right eye is slightly different. You can demonstrate this to yourself by staring at an object in front of you. It could be a chair or a table, what have you. And if you close your right eye, what you'll see is a certain amount of visual information coming in. And then if you turn around and you close your left eye, one of the things that you'll notice is that it seems like the objects move, but they don't really move. What's happening is that different information is coming in each eyeball, and what happens is that the brain takes this information, integrates it as if it were a single image, and provides to the brain this idea of depth perception. Okay, the old maxim is eyes in front likes to hunt, eyes on the side likes to hide. This refers to the difference in the orientation of the eyes in a hunter-gatherer, like a man, or a carnivore, for instance, versus something with panoramic vision, like a horse or a deer. Okay, so this yields our depth perception. And if you don't believe me, put a patch over one of your eyes and try and play catch with somebody, and you'll find that it's extremely difficult to do. So this was a major step forward in evolution for man, because this allowed him to keep from being eaten and to know how far away a prey item was. So they're important components in survival. These are just some eye disorders that you want to become familiar with. Myopia refers to a problem known as nearsightedness. The focal point is too near the lens, so the image gets focused in front of the retina. These individuals have to be much closer than normal to the object they're looking at in order for them to see it very clearly. Hyperopia is farsightedness. The result is that the image is focused behind the retina, and the problem here is opposite. These individuals have to be very, very far away from the object they're looking at in order to see it clearly. Presbyopia is simply a condition of lens stiffness that occurs as we age. One of the things that happens as we get older is that the lens itself becomes less pliable. As a result, it's more difficult for us to focus on items near our eye, and so they become blurry. And this is one of the reasons why, as we age, sometimes we need reading glasses to change the focal length of the image so that we can put it right smack on the back of the retina. Astigmatism occurs when the corneal surface isn't perfectly smooth. It has divots and ridges in it, and the result is that the image that comes into the eye, well, some of it will come in too soon, some of it will come in too late, and the result is that the image will be blurry. We can correct this either with LASIK or with other types of laser corrective surgery, or we can use, in some cases, bifocals, or in some cases contact lenses. Strabismus is a lack of parallel light paths through the eye. Basically, what happens here is that you don't get an accurate depth of field. Okay, what will happen is that part of the image will come in and intersect other portions of the image, and this interferes with what you're trying to look at. Retinal detachment can result in complete blindness. Basically, if the retina pulls away from the back of the eyeball, what's going to happen when the light comes into the eyeball is that it's going to miss part of the rods and the cones. The result is that that information won't get back to the brain. Glaucoma is an increase in interocular pressure caused by a problem with the aqueous flowing back into the bloodstream through the canal of Schlemm. We can deal with the pain with painkillers, but we can also surgically correct in some cases. A cataract is a clouding of the lens where, due to a change in the orientation of the proteins that are in the lens, the crystallins can become disorganized, and the result is that when the light hits the lens, it'll scatter instead of bending. And the result is a cloudy lens. When you look at these individuals, you'll see essentially an opaque cast to the pupil. And sometimes you can also get a scattering of light in the cornea. Okay, the cornea, as a result of trauma or disease processes, can lose its transparency and also scatter instead of refracting light. Okay, so you can get something that's like a cataract in the cornea as well. Macular degeneration we've discussed. There's two types, wet and dry AMD. In wet AMD, what happens is that you bleed under the retina, and the retina lifts away from the back of the eyeball, and you lose that part of your visual field. And you can correct this using photocoagulation, where we zap a laser into the back of the retina and seal off those bleeding blood vessels. In dry AMD, what happens is tiny growths, benign growths called drusen, begin to increase in size between the retina and the rest of the back of the eyeball, and they push the retina away from the back of the eyeball, and you lose that part of your visual field. I mean, we can sometimes correct this surgically, sometimes we can do something with lasers, and sometimes we can't do anything about it. Diabetes, of course, is a problem with the control of blood sugar, and what often happens in diabetics is because of the increased amount of glucose in the blood pressure tends to rise, and you run a great risk of bursting capillaries, and capillaries nourish the retina. If you burst those capillaries and bleed out, then the retinal cells can die and you can have early onset blindness. This is one of the early indications of problems with controlling blood sugar. We call this diabetic retinopathy. Okay, next special sense we want to talk about is hearing and balance. Your external, middle, and internal inner ear make up the three parts of the auditory system. It's also in part responsible for your ability to maintain your balance. The external ear terminates at the eardrum and begins at the pinna. Okay, this is the pinna, this is the external auditory canal, and this is the tympanic membrane or eardrum. The middle ear is an air-filled space between the tympanic membrane and the structures of the inner ear, which include the cochlea, the vestibular apparatus, and the semicircular canals. And the inner ear, of course, is responsible for our sense of hearing. That's the cochlea at work, and our balance, which is the vestibular apparatus and the semicircular canals working together so that we can stand up straight either when we're still or when we're moving. Okay, so this provides a lot of important information that allows us to maintain our equilibrium. If we have a problem with the functioning of the cochlea, we can have deafness or at least loss of certain ranges of our hearing. If we have problems with the vestibular apparatus or with the semicircular canals, we have trouble standing up straight. Okay, you're going to have trouble maintaining your balance. The external ear contains the auricle or pinna, which is elastic cartilage covered with skin. Its design is so that it funnels sound into the external auditory canal so that it can head towards the eardrum. The external auditory canal is lined with hairs and ceruminous glands which produce cerumen, which we also call earwax. The purpose of the cerumen is to keep out mold, to keep the skin of the canal supple, and also to inhibit insects trying to get into the external auditory canal. Okay, a variety of antibacterial components are also found in cerumen. A moderate amount of cerumen is good. If you have too much cerumen, you can occlude the auditory canal, and this can result in irritation and inflammation of the external auditory canal, which we call otitis externa, okay, or swimmer's ear. Too little earwax can result in promotion of the growth of mold. If the mold gets too far down into the ear, it can actually eat away the eardrum. You lose your tympanic membrane and you lose a great part of your hearing. The tympanic membrane is a thin membrane, 2 layers of epithelia with connective tissue in between. When sound waves hit it, it vibrates. It's the border between the external and the middle ear. Okay, it's very delicate. Okay, if you abuse it, it will rupture. Okay, and there's a variety of ways that you can abuse the tympanic membrane. And if it becomes traumatized and has to reheal, it can accumulate scar tissue, causing it to become less responsive to sound. The middle ear is separated from the inner ear by the oval and the round windows. These are very thin membranes. On top of the oval window is a little tiny bone called the stirrup, and the round window is essentially a pressure relief mechanism for the cochlea. Essentially, when sound comes into the ear, it gets focused by the pinna onto the tympanic membrane. This causes the ossicles to vibrate. The 3 ossicles are the hammer, the anvil, and the stirrup, but we want to know their formal names, and so that's the malleus, the incus, and the stapes. As the malleus hits the incus hits the stapes smacks on the oval window, and it causes fluid to move around inside the cochlea. That pressure is relieved at the round window, but as that fluid moves around inside the cochlea, it causes movement of very special cells in the organ of Corti, which have little projections called stereocilia on their apical aspect that essentially are very, very close, or in some cases embedded in a tectoral membrane. And when these little cells bounce up and down, what happens is that those stereocilia hit the tectoral membrane, they open up ion gates, and this is what creates the current that gets sent back through the vestibulocochlear nerve to the auditory cortex of the brain in the temporal lobe, and that's what we interpret as sound. There's 2 passages for air. The auditory or eustachian tube connects the pharynx to the middle ear, and this helps equalize pressure on the medial and lateral side of the drum. And there's also a passage to the mastoid cells in the mastoid process. The ossicles we've discussed, okay, we know what they're for. They transmit the sound from the tympanic membrane to the oval window. The oval window is again the connection between the middle and inner ear. The foot of the stapes rests here and is held in place by an annular ligament, okay, very similar to the annulus that surrounds the intervertebral disc, except much smaller and a little bit more delicate. Okay. Okay, here you can see the medial view of the middle ear. Okay, you can pick up the tympanic membrane. You can see the malleus. There's the stapes, and there is the incus. All right. There's the stapedius muscle, controls the movement of the ossicles, and that's the tensor tympani muscle, and that controls essentially it's a braking system so that in the event of loud sounds we don't abuse this apparatus to the point where it completely shatters. Okay, so it's protective for the drum and it's also protective for the ossicles. Okay, you can also see that there are ligaments that attach the bone of the middle ear to the temporal bone. Okay, and you can see branches of, again, the vestibulocochlear nerve running through here as well. Okay, again, that's going to be coming out of the cochlea and the vestibular apparatus. This nerve here that you can see in the photograph, or in the drawing rather, is the chorda tympani nerve. Okay, so it's responsible again for bringing sensation from the structures that it innervates, but it also talks to the apparatus in the middle ear as well. Okay. Let's talk a little bit about the inner ear. The inner ear is embedded in the temporal bone. The 2 labyrinths are the bony and the membranous labyrinth. The bony labyrinth is basically a series of canals inside the temporal bone housing the cochlea, the vestibule, and the semicircular canals. The membranous labyrinth are the tunnels and chambers in the bony labyrinth that are made up primarily of soft tissue and are part and parcel of the organs, both of hearing and of balance. There's endolymph in the membranous labyrinth, and there's perilymph in the space between the membranous labyrinth and the periosteum of the bony labyrinth. Okay, so you got fluid running through both of these structures. You got fluid running inside, and you've got fluid running around the outside of these structures in both cases. If we look at the inner ear, the oval window communicates with the vestibule, which communicates with the scala vestibuli of the cochlea. The scala vestibuli extends from the oval window to the heliocentrema at the cochlear apex. Notice that the cochlea has kind of a snail shape to it. The broad end, okay, is going to be near the point where the stapes attaches to the oval window, and the narrow end is going to be towards the center of the cochlea. And different regions of the cochlea, as we're going to find out, respond to different wavelengths of sound. Second cochlear chamber, known as the scala tympani, runs from the heliocentrema to the round window. Remember that the round window is that pressure release valve that allows the fluid to move inside the cochlea, but does not allow the organ to become damaged. If there were no pressure release valve for the fluid inside the cochlea, then when the stapes pounds on the oval window, you could potentially rupture the organ. Okay, so you got to have somewhere for the fluid to go, and that's what happens at the round window. The scala vestibuli and tympani are filled with perilymph, okay, and the endolymph is deep to that. Okay. Okay, the wall of the scala vestibuli is the vestibular membrane. The wall of the scala tympani is the basilar membrane. The cochlear duct, known as the scala media, is the space between the vestibular and the basilar membranes, and these are filled with endolymph. Okay, remember the endolymph is deep to the perilymph. The width of the basilar membrane increases from 0.4 millimeters near the oval window to half a millimeter near the helicotrema. Near the oval window, the basilar membrane responds to high-frequency vibrations, and near the heliocentrema, we respond to the low-frequency vibrations. So what this is a cutaway showing you now the structures that are in the organ of hearing, the organ of Corti. And what you can see are the outer and the inner hair cells, which are affixed to the apical portion of the cells that are responsible for responding to hearing. Okay, these are the hair cells. They're not hairs like hairs in your head. The hairs refer to the stereocilia that protrudes from them. Okay, but the part of the organ of hearing that actually generates the current that gets sent back through the vestibulocochlear nerve to the brain primarily comes from reaction of the inner hair cells hitting the tectoral membrane and opening those ion gates that are attached to them, causing a change in the current. Okay, so this is a branch of the vestibulocochlear nerve. This will eventually head back to the temporal lobe of the brain, and this is how we get our sense of hearing. Okay. Okay, the spiral organ, which is the organ of Corti, are the cells that are in the cochlear duct. The hair cells have hair-like projections at their apical ends. These are the stereocilia. The basilar region of the hair cells is covered by the synaptic terminals of the sensory neurons, while the cell bodies of the afferent neurons are grouped into the cochlear ganglion, also called the spiral ganglion. The afferent fibers form the cochlear nerve, and here we can see a zoom up showing you exactly how the inner ear does its trick. Hair cells are arranged in rows, okay, the basilar membrane on the bottom, the tectoral membrane on the top, and the hair cells between them. Okay. There are inner hair cells that are responsible for our hearing, and the outer hair cells, which are essentially there to regulate the tension on the basilar membrane. Hair bundles are the stereocilia of one inner hair cell. There are tip links which attach the tip of the stereocilia in a hair bundle to the side of the next longer stereocilium. So they kind of are like dominoes hooked together in a sense. As the stereocilia bend, they open potassium gates, which are mechanically gated ion channels. That's where the inner hair cell produces the current that we interpret now as sound. Okay, so here we get the mechanics of all this. In an unstimulated cell, the tip link is relaxed and the potassium gate is closed. As the stereocilia are bent towards the taller stereocilia, and this happens because, again, the endolymph is moving, causing the basilar membrane to move up and down, and that's what causes these cells to tilt. Okay, what happens is the tip link will stretch. The force created by stretching the tip link opens the gate of the potassium channel. In comes the potassium, and this causes a change now in the membrane potential, and this is what gets sent back. Okay, what you're looking at here is just a slide showing you what sort of form of energy sound is. And sound is a pressure wave. It's propagated through a gas, and it's the result essentially of gas molecules colliding with each other. Vibrations produce these sound waves. The volume or loudness is going to be a function of the amplitude of the wave, which is the height of the sound wave, while the pitch is going to be a function of the frequency, and the timbre is essentially a resonance quality, which comes from primarily the combination of different sound waves with each other, which can either add or interfere or partially interfere depending on the phase of the wave. You know that when a wave is propagated, it's going to have a peak and a trough. You also remember the definition of wavelength, and if we were to show wavelength here, essentially goes from peak to peak, okay? And this distance is defined as lambda, which is the wavelength. And again, remember that the number of waves that pass a given fixed point per unit time, pick some point, some physical point, indicated by the circle. The number of waves that goes past there per unit time is the frequency, and it's indicated by the Greek letter nu. Okay? So bottom line is the higher frequency sound you have, the higher energy you have in the wave, for the same reasons that apply to electromagnetic radiation. Now what you're looking at here is a little video that talks about the effect of sound waves on the cochlea. So take a listen.
Sound waves strike the tympanic membrane and cause it to vibrate. Vibration of the tympanic membrane causes the three bones of the middle ear to vibrate. The footplate of the stapes vibrates in the oval window. Vibration of the footplate causes the perilymph in the scala vestibuli to vibrate, which in turn causes displacement of the basilar membrane. Short wavelengths from high-pitched sounds cause displacement of the basilar membrane near the oval window. This movement is detected by hair cells of the spiral organ, which are not visible in the animation. Long wavelengths from low-pitched sounds cause displacement of the basilar membrane far from the oval window. Again, this movement is detected by hair cells of the spiral organ, which are not visible in the animation. When the vibrations reach the perilymph in the scala tympani, they travel to the round window where they are dampened.
Okay, there are several muscles in the middle ear that help to stabilize the ossicles and also to some extent the drum. The tensor tympani muscle inserts on the malleus and is innervated by the fifth cranial nerve. The stapedius inserts on the stapes and is innervated by the seventh cranial nerve. And they're responsible in part for the attenuation reflex, where the muscles will contract when there's loud noises to prevent damaging vibrations. This is so that you don't fracture the ossicles, and to some extent so that you don't rupture the drum. So these are safeguards that sort of keep that from happening. What they'll do is they'll tense up in the event that extremely loud noises are coming down the pipe. If you've ever seen the ossicles, and you'll get a chance to look at them in lab, they're some of the smallest bones in the body. You need really a magnifying glass to see them in any kind of real detail. This is just a diagram showing you how different parts of the cochlea respond to different frequencies and wavelengths. So what you'll notice here is that the higher pitched sounds are going to be picked up on that portion of the cochlea that is nearest the oval and round windows, and the lower frequencies, the lower pitches, are going to be picked up closer to the apex of the organ, which looks like the center of the snail shell from the outside. And the reason for this is that the properties of the basilar membrane are different as you go from the apex of the organ all the way to the portion that's nearest to the oval and round windows. Okay, the tension in the basilar membrane is a property of the tissue that composes it. More than 90% of the afferent axons of the cochlear ganglion synapse with the inner hair cells, and this is about 10 to 30 hair cells per ganglion. A few tiny diameter afferent axons synapse with rows of outer hair cells, which receive efferent input and cause them to shorten. Remember that they're primarily responsible for adjusting the tension in the system of stereocilia that eventually is going to produce the change in polarization in the hair cell, in the inner hair cell, that's going to lead to the production of a potential that could become an action potential and lead to transmission of an electrical impulse to the auditory cortex of the brain, which is in the temporal lobe. These hair cells that go all along the spiral organ are tuned to specific frequencies. There's actin filaments in the hair cells that attach to potassium-gated channels and move them along the cell membrane and tighten or loosen the spring. The hair cells are tuned again to very specific frequencies. Localization of pitch along the cochlea is a part of this property. The afferent cochlear nerve fibers send action potentials to superior olivary nuclei in the medulla. Okay, remember that those are a pair of posterolateral bodies. If you look at the pyramids, they're just behind and lateral to the pyramids. These are compared to one another, and the strongest is taken as the standard. The efferent action potentials inhibit other action potentials. Action potentials from the maximum vibration are going to end up going to the cortex, and that's what gets perceived as sound. And what you're looking at here is just the pathway that sound takes to get to the brain. So sensory axons from the cochlear ganglion terminate in the cochlear nucleus in the brain stem. Axons from the neurons in the cochlear nucleus project to the superior olivary nucleus or to the inferior colliculus, and axons from the inferior colliculus project to the medial geniculate nucleus of the thalamus. Remember the thalamus's role as the brain's sensation router. Okay, it basically directs input that comes from the brainstem to the appropriate area on the cerebral cortex, so that we can then assign that sensation a category. The thalamic neurons are going to project to the auditory cortex in the temporal lobe, and neurons in the superior olivary nucleus send axons to the inferior colliculus, back to the inner ear, or to the motor nuclei in the brainstem that send efferent fibers to the middle ear muscles. And again, that's going to come into play primarily when you experience extremely loud noises. But this also in part adjusts the tension on the drum and controls the amount of vibration in the ossicles so that it's optimized for the conduction of sound, but also so that you avoid damage to those structures. Okay, the next set of organs in the inner ear we want to talk about are those that deal with our sense of balance. The static labyrinth is made up of the utricle and the saccule in the vestibule, and they evaluate the position of your head relative to gravity and detect linear acceleration and deceleration, just as you would be able to pick those up in a car if you were to go from a stop to a steady velocity, or if you were to suddenly put on the brakes. You would pick that up as a change in the rate of movement, and then what would happen is that you would feel sensation as a force. Okay. The kinetic labyrinth is made up of the semicircular canals, and they evaluate movement in the head in three-dimensional space. So as you move left, right, up and down, or as you rotate, the fluid in the semicircular canals is going to move as you move your head, and that will report to the brain via the stereocilia, which in this case are embedded in a gelatinous cupula, which moves as fluid rushes past it, and that's what produces information about the orientation of the head with relation to gravity when it's moving in space. The static labyrinth and the utricle and saccule are similar, except that the stereocilia here are embedded in a gelatinous matrix that contains otoliths, little crystals, that respond to gravity. So when you're standing still, or when you're moving in a straight line in any direction, it's going to respond to gravity, and that'll tell you essentially what's down and what's up. And what you're seeing here is just a look at the static labyrinth and how it's set up. The utricle has a macula that's oriented parallel to the base of the skull, while the saccula has macula oriented perpendicular to it. The macula are specialized epithelia of supporting columnar cells and hair cells that have stereocilia and one kinocilium embedded in a gelatinous mass that has the otoliths in it. The mass moves in response to gravity and bends the hair cells and generates an action potential. The otoliths stimulate the hair cells with varying frequencies. The patterns of stimulation are translated by the brain into information about head position and about acceleration. Okay, so again, it's gravity that's primarily at work here. The gravity moves the otolithic membrane. The crystals, the otoliths, give it weight, and this bends the kinocilia. They produce now a potential. That potential is picked up by nerve fibers in the vestibular branch of the vestibulocochlear nerve, and that gets relayed back to the balance centers in the brain, which are in a different location than the auditory cortex. And what you're looking at here is just the mechanism showing you how the vestibule operates in maintaining balance. So as you move your head, okay, the endolymph in the utricle moves, okay. The otolithic membrane responds to gravity, okay, and that force of gravity is what's going to move the hair cells, primarily their apical cilia. That's what produces the current via a generator potential, and that's what gets transmitted to the vestibular nerve of the vestibular branch of the vestibulocochlear nerve. Now, the kinetic labyrinth is responsible for transmitting information to the brain as you move. Okay, so it's designed to do one thing, which is to respond to acceleration. All right, so there's three semicircular canals filled with endolymph. There's a transverse, a coronal, and a sagittal plane for each of them, and you can see them oriented to here. Okay, those are the three semicircular canals. This is, of course, embedded in the temporal bone of the brain. Each base is expanded into the ampulla and it has a sensory epithelia they call the cristae ampullaris. The cupula is that gelatinous mass that the cilia are embedded into, okay, the cilia that project from the apical surface of, again, the hair cells here. And it's a float that gets displaced by fluid as it moves past it in the semicircular canals. The displacement of the cupula is intense when the rate of head movement changes, thus the system detects acceleration rather than simple movement. Okay? And what is acceleration? Acceleration is essentially a change in your velocity. Okay, that's what we define acceleration as. So as you go from not moving to moving, you would define that as acceleration. As you go from moving to not moving, we would define that as deceleration, and the kinetic labyrinth would pick that up. In addition to that, any movement in a circular direction is defined as acceleration due to the fact that when you're engaging in circular motion, you're constantly changing the vector of that motion, and that satisfies the physics criteria for acceleration. Okay. So what are the pathways that this takes to the brain? Well, most of these centers are found in the brainstem and in the cerebellum. And this is important because remember the cerebellum is responsible for coordinating our intentions with our actions as far as movement is concerned. So what you have is sensory axons from the vestibular ganglion passing through the vestibular nerve to the vestibular nucleus, which also receives input from several other sources such as proprioception from the legs. Vestibular neurons send axons to the cerebellum, and this influences your postural muscles. The vestibular neurons also send axons to motor nuclei, primarily the oculomotor, the trochlear, and the abducens. And remember, what those are responsible for is moving the eyeball and the eye socket. So this allows our gaze to track with our movements. The vestibular neurons also send axons to the posterior-ventral nucleus of the thalamus, which in turn projects to the vestibular area of the cortex, which lies in the parietal lobe. Okay, remember that that's the lobe of the brain that lies posterior to the frontal lobe and is essentially defined by the central sulcus being the border. Okay, so this is what allows us, again, to compensate for our movements and also at the same time to fix our gaze despite the fact that we're moving. I know it's kind of weird to think about, but while you're listening to this lecture, look at something across the room and move your head. And as you move your head, you can keep your gaze fixed on an object and the object won't move. You're doing that somewhat subconsciously, and that's due to the fact, in part, that these vestibular reflexes are responsible for or are coordinated with our eye movement. If you purposefully lock your gaze so that as your head moves, your eyeballs move, okay, you can do this consciously and override this reflex, then as you move your head, everything is a blur, okay, and it gets very difficult for you to see where you're going. All right, so this is designed essentially for our survival, very primitive reflex, and part of what goes into this are the actions of the inferior colliculi, okay, that are going to control our eye reflexes in response to our movement. So as we age, of course, our special senses don't improve. Virtually none of our physiology improves as we age. We see a loss in the ability to detect odors. We have a decreased sense of taste. Our lenses become less flexible. We suffer from presbyopia, so it's hard for us to focus near. And we see the development of cataracts. We also experience macular degeneration, glaucoma, and diabetic retinopathy. And we also see a drop in visual acuity and color perception. And that's inevitable. A lot of it is degeneration due to use, but some of it is just due to the fact that Again, the components of these systems naturally will begin to die as we get older because they have a finite lifetime. Part of that is determined by the amount of energy they get, which is in part due to their blood supply, but part of it is due to an intrinsic clock that essentially runs out, and what happens is that as cells begin to die, tissues begin to die. As tissues begin to die, the organ function is compromised, and eventually the organ system will shut down. Okay, that concludes this portion of the lecture. Again, study this material as it will be on the PRS quiz, and I will see everybody in class. Thank you.